So you may be wondering, why would I want to publish my work in PEDS? So I'll describe a few benefits of doing so. So the first one is that we recently moved to a online-only continuous publication model, which expedites the time it takes for your paper to become available to the community after it gets accepted. And this has also enabled us to get rid of all publication fees except for the optional open access fee. That means that if you publish in PEDS, it's not going to cost you anything. And this is unlike some other journals who are society-run journals that always charge you a certain amount to finance the society. Remember that PEDS is a community-run journal, so we don't have a society to maintain. We also, another benefit is that, probably the most important, is that we have an editorial board structure that contains leading experts in the field. And this ensures that you get a fast review, also that is rigorous yet fair. And we also strive to make it transparent to get you the feedback you need to make your work even better. And recently, I've updated the editorial advisory board to be more diverse, to include a diverse group of experts from different geographical locations, different backgrounds, with different research expertise, as well as at different stages in their career. And the goal of having a diverse editorial advisory board is to help me steer the journal to meet the needs of our community. Now, even though we have this editorial advisory board structure, I want everyone here to feel comfortable contacting me or anyone at the journal if you have comments or suggestions to help make the journal better for you. Remember, we are a community-run journal, so we're here to serve. We want this journal to be the best it can be for everyone here. So with the final few minutes, I'd just like to mention a few exciting initiatives that we have in the works. So the first one is a reviews special issue that should be coming out in the second quarter of 2021. And it's going to be really exciting because these reviews will focus on current topics. So it'll be focused on the last few years of what came out in the last few years. And there's a wide range of different topics. So there should be something for everybody here in that issue. And importantly, all of these reviews will be made freely accessible to everybody. That means that you won't have to have a subscription to the journal to be able to read them. Secondly, we're also creating an annual award for the best paper published in the journal. And this award will be given to the junior scientist who's responsible for the work. So that means the graduate student or the postdoctoral fellow that did the experiments. And the purpose of this is not only to promote the journal, but also to help in the career development of junior scientists and to help them promote their work. Because I think it's our responsibility as a journal, but also as senior and established researchers to help the next generation of protein engineers. And lastly, the final initiative that I want to mention are these webinars that are starting today. So this idea of organizing webinars came about given the current pandemic situation where we couldn't physically meet at our usual conferences and learn about the recent results. So I figured it'd be nice to get everybody together online, hear about the latest and greatest research, and if there is interest, we'll keep organizing these webinars beyond the first 4 that we've organized this fall. And you may have noticed from the ad that the 4 speakers, the keynote speakers, are all internationally recognized leaders in our field, so it should be very exciting to hear about their latest work. But also importantly, all of these webinars will include a presentation by a junior scientist who will present their work published in the journal. And again, the idea is to give them a platform to promote their work, and this should be helpful in their career development. So that's it for me. I'd just like to give a couple of details about how the webinar will work. So first of all, we're going to start with a short presentation from a junior scientist who will present their work published in the journal. And that work was selected as the editor's choice article, so you can read it for free on the journal. So we'll have the presentation followed by a question period. And if you want to ask questions at any point during these presentations, you can use the Ask a Question button at the bottom of your window there. Or if you're on YouTube, you can type in the chat box your question. And these questions will be read and answered by the speaker. And even if we run out of time in the question period and there are some outstanding questions left to be answered, the speakers have agreed to answer them offline. So you will get your answer in the text box. After that, we'll follow with our keynote presentation, which should be about 40 minutes. And there will also be a second question period after that presentation as well. Okay, so that's it for the the explanation of the webinar and the journal. So I'd like to now introduce our first speaker of the day. So our first speaker is Dr. Joshua Atkinson, who's currently an NSF postdoctoral fellow in the group of Mo El Nagar at the University of Southern California. And he's going to present his work that he did during his PhD with Josh Silberg at Rice University. And he's going to tell us about circular permutation and its effect on stability. So please take it away, Josh.
Let's get the screen up here. Hopefully folks can see that. Alrighty, good morning, and thanks for that introduction. And I'd like to thank Roberto Chica for selecting our article as the editor's choice and giving me the opportunity to present our work. Today, I'm going to tell you about our efforts to see if deep mutational scanning experiments can yield information about a protein's tolerance to topological mutations and changes in conformational flexibility. Proteins are dynamic macromolecules that oftentimes remodel their intramolecular contacts to adopt different conformations that are critical for their functional activity. But it's often challenging to predict where within a protein that conformational dynamics are going to be important for function. And this limits our ability to design new functional proteins from the ground up. But one approach to identify regions of proteins where altered flexibility can impact function is using mutagenesis experiments. Recently, deep mutational scanning has emerged as a powerful technique for comprehensively sampling mutations in a protein. In a mutational scanning experiment, a combinatorial library of mutant proteins is generated, and then they're enriched for functional variants using a growth selection or a means to physically separate cells like FACS. And then these variants are scored using DNA sequencing. And this has enabled many individual sequences to be linked to their fitness in a single experiment, generating high-resolution maps of sequence function relationships. But what types of mutations can we use to alter local flexibility within protein structures? Mutational scanning experiments have been applied to comprehensive amino acid substitution libraries, as well as libraries that alter the protein's topology through things like domain insertion, truncation, or circular permutation, where a sequence is reshuffled and N and C termini are generated at different positions within that sequence. Substitution experiments can allow us to perturb local dynamics through point mutations to more flexible amino acids like glycine or serine. But circular permutation can also perturb that conformational flexibility by replacing the peptide bond between neighboring residues with new N and C termini that are free to move about. But the global tolerance of circular permutation to proteins is less well understood than amino acid substitutions. So we were interested in exploring the impact of this type of mutation. So previous studies have illustrated that the parental protein's thermostability correlates well with its average tolerance to point mutations. So we hypothesized that this would hold true for circular permutation as well, where highly stable thermophilic proteins on average will tolerate permutation, the disruption caused by permutation, relative to more marginally stable cyclophilic variants that are on edge with whether or not they're folded proteins. And we also wanted to see if we were to scan multiple protein orthologs with differing thermostabilities in parallel with one another, if that would allow us to tease out some local patterns of mutational tolerance that might inform us about the conformational dynamics of the protein. And the reason why we thought that might be the case is because proteins evolve in an energy landscape that is constrained by the need to minimize their free energy of their contacts so the protein can fold, while still maintaining sufficient local configurational entropy so the protein can traverse the conformational landscapes necessary for its function. And this trade-off is evident when you look at proteins that have adapted to different temperature niches, with cyclophilic proteins adopting more molten, flexible structures and thermophilic proteins becoming more rigid to compensate for the effect of temperature on their dynamics. One way to go about identifying regions within the protein that might have evolved to increase the local configurational entropy is to look at the energetics of the contacts within that protein. So residues within a protein that have been selected by evolution to minimize their energy of their contacts, in the language of energetic frustration, can be called minimally frustrated residues. And this means that the amino acid that was selected at that position is best suited for minimizing the energy of the contacts necessary for that protein to fold. While some residues have amino acids that display high energy contacts relative to other amino acids that could have been selected for at that position. And these can be described as being highly frustrated. Minimally frustrated residues have been found to be important for stabilizing protein structures, and while highly frustrated residues have been found to be localized near functionally important regions of the protein, including binding interfaces and enzyme active sites. So in our study, we wanted to determine if these local energetics might influence the tolerance to permutation, which will increase the local conformational flexibility. And we hypothesized that minimally frustrated residues of the protein would be less tolerant to mutations than highly frustrated regions of that protein. And that's because the local contributions to the free energy of folding are greater for these minimally frustrated locations than they are for these highly frustrated locations. So when permutation disrupts that contribution to folding, it will be less severe for the highly frustrated than it will be for the minimally frustrated. We wanted to see if this trend might become more evident when you scale the global thermostability of that protein up and down in a mutagenesis experiment. So we selected adenylate kinase as a model protein for exploring this interplay between stability and functional dynamics. This enzyme is essential for maintaining energetic homeostasis inside the cell by converting ATP and AMP into ADP, and has been extensively studied over the years, over the course of its entire catalytic cycle. Adenylate kinase consists of 3 domains: a core domain, a lid domain, and an AMP binding domain that during their catalytic cycle undergo a series of conformational shifts. Initially, the protein can be found in an open resting state, and upon substrate binding, the lid and AMP binding close down onto the core domain, positioning the substrate near the active site that is already pre-organized for rapid phosphotransfer. And then the rate-limiting step of this catalytic cycle is the reopening of that AMP binding domain to release the products from the enzyme. And these 3 domains have evolved to independently tune different aspects of the catalytic cycle of adenylate kinase. The core domain has evolved to minimize its conformational dynamics and controls the global stability of this protein, while the lid domain has evolved unique conformational dynamics within that domain that have been shown to tune the protein's affinity for ATP. And similarly, the AMP binding domain has evolved conformational dynamics that enable it to tune the product release and enzymatic turnover of adenylate kinase. So we wanted to see if these domains that have evolved for different conformational dynamics would differ in their tolerance to circular permutation. So we selected 3 different adenylate kinases that have thermostabilities varying over 30 degrees to generate different permutation libraries. And these had all had solved structures and had been previously studied. And then to build our permutation libraries in a systematic way, we use transposon mutagenesis to generate this library. So we use a synthetic transposon that we call a permuteposon that contains all the genetic elements required for a protein expression vector, so an origin of replication, a promoter, RBSs, and an antibiotic resistance gene. And then we use the mu-A transposase to insert this transposon randomly throughout a circularized adenylate kinase gene. And then to maintain the uniform translation initiation and protein expression of the different mutants in our library, this transposon also encodes an 18 amino acid N-terminal tag that maintains the genetic context near the ribosomal binding site. So when transposons insert into sequences, they do so in 2 orientations at equal frequencies. So one of these orientations is a parallel orientation where the regulatory elements of that expression vector are in the same direction as the coding sequence of our protein mutants. And the other orientation is an anti-parallel direction where these regulatory elements are the opposite direction of the coding strand. And because transposon insertion frequency is not uniform throughout the entire adenylate kinase sequence, we use these anti-parallel sequences as internal negative controls because they're unable to express proteins. And this allows us to calculate statistics regardless of the initial frequency of each mutant within our library. So the next thing we did is we took these libraries of circularly permuted AKs and we put them into a strain of E. coli that contains a temperature-sensitive adenylate kinase, and it's unable to function at higher temperatures. We then grew these cells under a selective condition at 42 degrees Celsius, as well as a non-selective condition under 30 degrees Celsius, where that endogenous adenylate kinase is functional. Then we harvested the DNA and then sequenced it using MySeq and used a computational pipeline to generate circular permutation profiles. So this is where we quantified the fold enrichment of each variant between the selective and non-selective condition. So you can see here in positively enriched sequences, I'm showing here in blue, while negatively enriched sequences are being shown here in gray. So next I wanted to show you the permutation profile for our cyclophilic adenylate kinase, so the cold-loving, more molten protein. So this variant, just to walk through what this means, if variants were found to be statistically enriched relative to those anti-parallel variants I mentioned, they're shaded in this gray to black color. So these are functional or marginally functional proteins within our library, while those that are found not to be statistically enriched relative to those anti-parallel variants are shown here in red, and these are non-functional protein variants. And then these little black bars down here at the bottom are regions of the protein that the transposon did not insert into and we did not sample. So as you can see, for the cyclophile, nearby the original N terminus and the original C terminus was tolerant to permutation and yielded functional mutants, as well as within that mobile AMP binding domain that I was talking about earlier. So now if we look at the mesophilic version of this protein, you can see there's a larger number of functional variants that were found in this library, and they're peppered throughout the core domain as well as the lid and AMP binding domain. And just if we increase the thermostability a little more to that thermophilic variant, you can see even more variants are now found to be functional within this library. Just to more clearly illustrate this point, we plotted the fraction of functional variants within our entire library relative to the parental thermostability of each of the adenylate kinases that we used. And this revealed to us that the ensemble tolerance to permutation correlated with the parental thermostability as we had expected based on studies of point mutation. So the next thing we wanted to see was, can we get family level trends about the local regions that can tolerate permutation within adenylate kinase? So to go about doing this, the first thing we needed to do was convert those fold change measurements that I'd mentioned earlier into a measure of protein fitness that we can compare between the three libraries that we had generated. And the reason why we needed to do that is our library also encoded for the native adenylate kinase sequence in addition to all of the mutants. And as the fraction of functional variants increased, the fold change enrichment of these proteins decreased. And this is just because of a competition for resource for more of these cells growing in the culture conditions that we were growing them in. So in order to normalize each per mutant's fold change to generate a fitness score, what we did is we just normalized that by the fold change of the native proteins within these libraries to get a measure of fitness. So if we look at the average fitness of each variant compared to the primary structure of adenylate kinase, you begin to see a trend for which domains tolerate permutation. So originally near the N terminus, you can see the wild type has wild type level fitness of 1. So this is wild type level of activity, this dotted gray black bar here. And then as we enter the core domain of this protein, quickly it no longer tolerates permutation. And then we can enter into the AMP binding domain, and you can see there's a rise in the tolerance to permutation up until this point where we actually begin to exceed wild type levels of activity in this protein. And in all three orthologs, this protein was found to be significantly enriched. And you can see that by the color of these circles here. Then as we enter back into the core domain of this protein, it's again no longer tolerant, except for in some regions where it's marginally tolerant. And into the lid domain, that's also true, where there's some marginal tolerance, and some that get near to wild type levels of activity. And then at the near the C terminus of this protein, more of the core domain tends to be tolerant to permutation. And to see these domain level trends a little more clearly, what I did next was just to plot the average fitness versus the number of orthologs in which it was found to be significantly enriched. So you can see here the intolerant positions that were not significantly enriched in any of the orthologs were predominantly found within the core domain, with a couple positions within the lid and AMP binding domain. And then as it becomes more tolerant in 1, 2, or 3 of the orthologs, you can see the average fitness increases, and more of the lid and AMP binding domain become larger populations. And for the most tolerant and highest fitness, these variants were found within the AMP binding domain. Just to show this one other way, we went ahead and plotted this average fitness data onto the structure of the protein. So all the positions in white here are the regions where 1 or more of the orthologs is unsampled within our library. So we didn't want to make any average fitness measurements on less than 3 of the variants. But you can see here that near the core of this enzyme, it tends to be have low fitness and is intolerant to permutation. While out in this AMP binding domain, it's exceptionally tolerant with wild type levels of activity. This one position in the lid domain is also pretty tolerant to permutation. And then the N and C termini are also showed tolerance, which you would expect since you're simply slightly adjusting where the terminus is. So what makes this AMP binding domain unique? So going back to the idea of energetic frustration that I was talking about, what we did is we calculated the density of frustrated contacts within adenylate kinase in both its open conformation, as shown by the dotted lines, and its closed conformation, as shown by the solid lines. And we did this using the frustratometer, a tool from the Woolness lab, who's also at Rice that we were discussing this project with at the time. So as you can see here, the region that has the highest concentration of frustrated contacts is within this AMP binding domain, and especially evident when it's in the closed conformation where the enzyme is clamped down on itself. There's also pockets of frustration found within the lid domain as well as in the core domain. And this can be seen a little more clearly if you look at the three-dimensional structure, where I'm showing the contacts that are minimally frustrated in green and the highly frustrated contacts in red. You can see this large cluster that is in this loop region of the AMP binding domain. So we plotted that average fitness versus this density of frustrated contacts in that closed conformation, and that revealed a positive correlation between these two values. And you can see here the AMP binding domain really stands out as having a large contact density of frustrated contacts and a very high average fitness relative to other permutants within our library. So just to summarize what we found in this study, there's domain-specific tolerance to increases of conformational flexibility caused by the permutation that we did. So if you permute within this core domain, it's often deleterious to the function of the enzyme, while when you permute in the lid domain, it can have intermediate effects. And then in the AMP binding domain, it's rarely deleterious to the activity of this enzyme. And just to conclude, we found that the average tolerance to new termini correlates with parental thermostability, just like it does with point mutations. The local tolerance to new termini has a correlation with the energetic frustration of the contacts within that region. And that combinatorial experiments where we look at multiple orthologs can help reveal family-level trends in mutational tolerance. And with that, I'd like to thank the Silberg lab, especially Alicia Jones, who started off this project and did many of the experiments, as well as our collaborator, Vikas Nanda at Rutgers, and our funding sources. And with that, I'm happy to take any of your questions. Thanks. Let's see here.
Okay, great. Excellent presentation, Josh. Are there any questions? Please use the text box, the Ask a Question text box, or the chat box on YouTube. Okay, so give a few minutes there. People tend to be shy. But maybe I'll start, Josh, with a quick question. It's very interesting that the AMP binding domain is the one that tolerates circular permutation. Do you think this has to do with the ability to bind and or release substrates and products? Like, did you look at effects on KM, for example?
We did not purify these enzymes and look at effects on KM, but I did, based off of just how this protein tends to function in its catalytic cycle. I want to go back here. During that last step where there's product release, what's thought to happen in the AMP binding domain is actually there's some local unfolding that happens that promotes it to pop open and release those products. And we've been thinking that this permutation enables that unfolding to happen on little quicker timescales. Yeah, but we haven't, we've been exploring this enzyme in vitro recently, but we haven't had any data yet on that front.
Okay, great, thanks. Okay, there's a question here for you. How does the tolerance to circular permutation correlate with flexibility of the region?
Yeah, so the reason why we've been saying that this increases the conformational flexibility is just because we've eliminated that peptide bond that physically links these two neighboring residues together, and then actually replaced it with charged residues that oppose one another that will then, have a somewhat of a steric hindrance with one another. So we don't have like NMR data to back up this claim of increases of flexibility, but there have been papers that have shown that changing this chain connectivity does alter the flexibility locally in those regions of those proteins, and that's why we make that claim.
Okay, are there any other questions for Josh? There's a lot of hands clapping, a lot of praise, not many questions, but that's good. I guess you were very clear. Okay, in any case, if you have additional questions, if you think of them in the meantime, please add them to the text box, and Josh, I'm sure, will be happy to answer them there.
Definitely. Thank you,
All right.
guys.
Thank you, Josh. Okay, so our next speaker is Professor Steve Mayo from Caltech. And it's a great pleasure to have him here today to tell us about his recent work. So Dr. Mayo has been at Caltech since 1992, and he's widely recognized as a pioneer in computational protein design, as being one of or the first person to design from physical principles and using a structure-based approach to design a protein to adopt a specific fold. And he's also, along with David Baker, a pioneer of computational enzyme design. So Dr. Mayo started his training at Penn State University, and then he moved to Caltech to work with Harry Gray. And during his PhD, he co-founded a molecular modeling company called Molecular Simulations, which eventually morphed into Accelris. And I think now they've changed names to something else. But basically, he helped develop a lot of the computational modeling tools that some of us have used. And after that, he also did two postdoctoral stints, one at Berkeley and one at Stanford, and joined the Caltech faculty in 1992. He quickly rose through the ranks. He was admitted into the National Academy of Science and was also a HHMI investigator. He also more recently dedicated a lot of his energy to administrative positions at Caltech. So he's been Vice Provost. He's currently the Division Chair of Biology. And we're very happy to have him here today to tell us about his recent work in computational protein design. So thank you, Steve, for joining us.
Hi, can you hear me? Great. So it's a great pleasure to be here. And I want to start by thanking Roberto for inviting me. I've been looking forward to this, particularly given the transition that I'm making from being division chair to getting back into the lab and focusing on research. I've been in an administrative position now for 13 consecutive years. That run will end on Monday, and I'm looking forward to doing some more interesting stuff on the computational protein design front. So today I wanted to tell you a little bit about some early work that we're doing in a new area for us, which is entitled CDR Extended Antibodies. So the basic idea is that we want to put a twist on to antibody-drug conjugates in a way that is, we think, interesting and potentially important for developing maybe future potential therapeutic molecules, but also just a lot of fun in terms of thinking about designing protein binding molecules. Okay, so the presentation outline, I want to just do a brief introduction to computational protein design using some slides that I've probably overused throughout the years, but I think they'll serve the purpose of putting everyone close to being on the same page. And then to talk about this idea of CDR extended antibodies, what the concept is, and then what our first test case is to try out the idea. Okay, so computational protein design is really sort of, you can think about it as a collection of different methods and then applying those methods to various applications. On the methods side, most computational protein design approaches require some sense of what the protein backbone is, require some description of the conformational flexibility of the amino acid side chains, which we capture as rotamer libraries. Of course, we're very interested in sort of quote unquote physics-based design approaches. And so in order to implement that, you need to have a atom-based molecular mechanics force field for computing energies. Then, of course, computational design is a combinatorial problem, and it's important then to have an approach to solving the combinatorial optimization problem. For many design problems, the issue of negative design is also very important. That is, it's not sufficient to design in a positive sense, that is, to design for your design objective. It's often important also to design against things that you don't want to have happen. For example, something like aggregation. Negative design is certainly has been and remains a significant challenge in the computational protein design field. So with this set of methods, you can envision applying them to a whole variety of applications, studying the sequence structure, stability, function relationships of proteins, thinking about the evolution of protein structure and function, various hypothesis-driven inquiries, and perhaps most interestingly, in terms of real applications, protein-based biotechnologies. And so many years ago now, we demonstrated that you can actually do all of this and computationally design novel amino acid sequences that actually fold with high fidelity to your target structure. We also demonstrated very early on that the optimization and the force fields actually matter because if you do it correctly, you could actually generate variants of proteins that are hyperthermostable to unfolding. And of course, the field has exploded over the last decades, and there are many labs working in this area, producing really quite phenomenal results from functional molecules to supramolecular structures, and it's been really impressive to see what's been happening over the years. So just to give you a sense of how the calculations are done, the sequence optimization calculations are done, I'll just go through this toy example. So we have a protein where we want to optimize two positions in the protein, P1 and P2. The first thing we need to do is define the set of amino acids and their rotamers for each of the positions in the design. So in this simple case, we have one alanine rotamer, 3 valine rotamers, and 3 serine rotamers at each of 2 positions. Of course, in a normal design calculation, there might be tens or hundreds of positions and hundreds to thousands of rotamers allowed for each position. The standard construction for computational protein design is to think about it as a problem that could be decomposed into pairwise interactions. And so there would be sort of 2 types of energies, a one-body energy and then a 2-body energy, and I'll get to that in a second. So for the one-body energy, it's the energy of interaction between the atoms of the side chain of the rotamer of interest at the position of interest interacting with the backbone template of the protein. And then, of course, we compute the energies using your favorite collection of potential energy functions that capture things like electrostatic energy, van der Waals, hydrogen bonding, solvent potentials as well. And then we compute this one-body energy for each of the rotamers in turn at each of the positions in the design calculation and store it as a vector. After computing the one-body energies, we then compute the 2-body energies by now ignoring the template, the backbone template, and looking at the interactions between the atoms of pairs of side chains, and then do that for all possible pairs of rotamers, and then fill out this matrix. So at the end of this setup calculation, you have a one-dimensional vector with the one-body energies and then a matrix with the 2-body energies. These can be pre-computed or for small problems, but for larger problems, we typically compute this matrix on the fly only as needed for the positions that are being considered in the design. Okay, then as far as optimization goes, there are many ways to optimize. In this simple case, what I've shown here is the two-body matrix and then the one-body self-energies broken into two pieces for the two positions. The simplest optimization protocol is using Monte Carlo, where we initialize the system using some random selection. So P1 randomly select serine rotamer 2, and then position P2 randomly select failing position number 1. Then you sum up the energies, the two one-body energies and the single two-body energy. And then using the Metropolis Monte Carlo procedure, you can make random moves and optimize the system. And in this case, the optimum energy, the optimal solution is easily found because it's a small problem, 2 serine rotamers with a total energy of -9 units. Then, of course, we can build that solution back on the protein and look at it and evaluate it for other features that we might be interested in. Many ways to do optimization. I'd say in the early days, optimization was a large concern, but given computer speed and the efficiency at which you can implement Monte Carlo algorithms, mostly everyone is using Monte Carlo for doing their simulations. Okay, so with that as an introduction to computational protein design, I want to turn to this idea of CDR extended antibodies. So the concept is a derivative of something that's been long studied in the drug industry, a molecule called an antibody-drug conjugate, an ADC. So an antibody-drug conjugate uses an antibody to target some typically a protein, usually a cell surface protein. That antibody then has a small molecule drug covalently attached to it with a PEG linker, for example, and the antibody drags that small molecule payload to some target, and then the payload is either released or can act while covalently attached to the antibody. It's a great concept. What it effectively does is it concentrates your small molecule drug in an area where it's most needed and potentially where it will have the least toxic side effects. So if you had a very toxic, very cytotoxic anti-cancer drug, you can minimize toxic side effects by concentrating the drug using the antibody on cancer cells. So importantly, the ADCs use, they target two independent sites. So the antibody has its own target, and then the small molecule drug has an independent target that it operates on. More recently, a group at Amgen came up with this idea of chemibodies. So this is an advance on antibody-drug conjugates that allows for binding synergy between the antibody and the small molecule binding to the target. So in this case, instead of two independent targets, there's a single target, but two independent binding sites. And because the small molecule is linked to the antibody, you get this synergistic effect in terms of binding. And in the Amgen example that was published a couple of years ago, they were able to get roughly, I believe, a 20-fold improvement in the affinity for the target. So what we're interested in is an evolution of this concept that we're calling CDR Extended Antibodies, that rather than targeting two different binding sites, we're targeting a single binding site. So there's one protein target and one binding site. And the antibody and the small molecule cooperate to bind the actual single binding site. And that's illustrated here on this slide. So in the Chemibody concept, this is from the Amgen group, published in 2018. The antibody has a PEG linker to a small molecule that binds to the active site of the target protein. And as you can see, the antibody itself, through its CDR loops, is binding the same target protein, but in a different region. Coupling the antibody to the small molecule covalently leads to an enhancement in the binding of the Chemibody to the target. In the CDR extended antibody concept, what we're doing is having also the small molecule covalently attached to the CDR, attached to the antibody through a position on the CDR loops. And then, of course, the small molecule binds, conceptually will bind to its binding site on the target. But in addition, we're building an interface between the CDRs and the protein target to effectively increase the binding surface area of the small molecule. So it's an extended CDR, chemically extended CDR, where we have now a much more extensive binding site for both the antibody and the small molecule. And the idea is that this will potentially enhance target specificity. It's a concept that should allow for the development of novel protein binders. And there are many potential applications. One potential application is to rescue failed drugs. So imagine, for example, that you have a small molecule drug that has shown efficacy in a particular clinical trial, for example, but then in a larger scale trial may have shown off-target side effects. There would be the potential to rescue the off-target side effects by increasing the specificity for the actual target, which in turn would decrease, presumably, the binding to any off-target. And so this, in some sense, adds an element of negative design that we're now, by enhancing the on-target binding through enhanced surface area interactions, we're decreasing the off-target interactions, and that could potentially rescue a failed drug, a drug that has failed because it's has off-target binding interactions. Anyway, we're very interested in this sort of concept, and we wanted to test out in a model system whether or not we could construct such a molecule and what its properties might be if, in fact, we could construct it. So we set out to utilize a fairly simple model system, which should be familiar to many of you, the binding of biotin to streptavidin. So in our target molecule will be a monomeric version of streptavidin, which has diminished binding interactions with biotin. We initially set out to use a triple mutant, a previously published triple mutant of streptavidin that both confers monomerization of streptavidin, but also reduces the binding affinity to biotin. For technical reasons, experimentally, we eventually had to switch to using a streptavidin derivative called MSA, MSA for monomeric streptavidin, which was much better behaved experimentally. Monomeric streptavidin, MSA, is very similar to the triple mutant that we were using initially, and I'll say more about that in a moment. For the small molecule, obviously we're going to use biotin, and in particular, we're using a C2 maleimide variant of biotin, where we can then use the maleimide moiety to couple to a cysteine that is judiciously targeted or positioned within one of the CDR loops. And then for simplicity for this model system, rather than using a full-length antibody or a Fab, we're using an antibody to make the experimental evaluations more efficient. Okay, so the design approach is the following. We first want to identify potential antibody streptavidin pairs. And of course, we want to do this computationally. And then we want to identify a CDR position on the antibody where we can introduce a cysteine that will then be the target for coupling to biotin. And then, of course, we want to optimize the protein-protein interface, the antibody-protein interface, and then any other region of the antibody that would require optimization in order to build a nice system. Okay, so what we settled on in terms of coming up with docking poses of the antibody against the streptavidin is to go to the PDB and look at a set of existing antibody crystal structures with antigens bound. And then the nanobodies that we selected for our particular study are shown here, aligned on the framework region. This is just 3 different views of the same set of alignments. The CDR loops are color-coded. CDR loop 1 is shown in red. CDR loops 2 are shown in yellow, and CDR 3 loops are shown in blue. And then we characterize the interfaces between each nanobody antigen pair in 3 different ways. The interface separation distance, the amount of buried surface area, and then we utilize a shape complementarity score. And so what's shown here are the averages and plus or minus standard deviation for the individual complexes. Okay, then we want to dock this library of nanobodies against streptavidin. Doing so, we generated 2,310 dock poses, which is too many to design on. And so we filtered them using the following criteria. So we looked at whether the nanobody, the putative nanobody streptavidin complex matched the interface statistics that we see for naturally occurring nanobody antigen interfaces. We, of course, wanted to only consider nanobodies that actually overlay the biotin binding site. So we had a metric for whether or not the nanobody blocked the binding site. And then we also were very interested in evaluating whether the putative binding of the nanobody to the streptavidin recapitulated some of the naturally occurring, the native nanobody antigen interactions that occurred in the particular crystal structure. And you'll see that that's, we were able to find poses that, that sort of optimize all 3 of these criteria. Okay, so applying the filters to the 2,300 dock poses, we ended up with 7 complexes that look promising. They're shown here. The nanobody is shown in cyan, and streptavidin is shown on the bottom in green. And you can see where the biotin is in all these configurations. We selected nanobody 4NBY, chain B, as our target complex to further elaborate. Okay, so then with a target complex in mind, we have to place the biotin and identify a position on one of the CDR loops for incorporating cysteine that will then allow for coupling to the maleimide. Now, of course, we know the bound configuration of biotin. So in our design calculations, we use the crystallographically determined binding of biotin. But of course, the tail of this modified biotin is unknown. And we developed a rotamer library to cover the conformational flexibility of the tail that would be exposed from streptavidin. And of course, we want to find configurations of this tail that then would allow for coupling the cysteine on a CDR loop. Okay, so so the biotin is anchored in the streptavidin in its crystallographic position. We utilized polyalanine CDRs just for simplicity. We implemented rotamer flexibility for the biotin tail, and then we evaluated for each rotamer steric clashes with both an antibody and with streptavidin, and then looked for proximity of the a putative C alpha position on the streptavidin molecule with a C alpha position with the correct stereochemistry on the CDR loop. Okay, so we were able to find a number of such solutions. We settled on position 103, which is in CDR3 of this particular nanobody. And you can see that the CDR3 loop extends over here. You can kind of make out the maleimide moiety, and it's hard to see where the connection is to the cysteine, but it's buried there. If we zoom in to the interface, what we'll see, this is the streptavidin on the bottom and the nanobody on top. There are two critical interactions between the CDR loop residues in the original nanobody antigen structure. So that's shown here on the right. A tyrosine making an interaction with a backbone carbonyl, and an arginine making also an interaction, with a threonine side chain. Those two interactions are recapitulated in our docked model, which is something we sought to achieve. So we have a tyrosine making a backbone interaction with this carbonyl, and an arginine picking up an interaction with a threonine side chain in a very similar configurations to what was observed in the actual crystal structure of the nanobody with its native binding partner. So that was encouraging. Okay, so I mentioned this problem with this triple mutant of streptavidin. In our hands, we were not able to express monomeric material that didn't aggregate. So we switched over to using MSA, monomeric streptavidin, which is an engineered version of streptavidin from another lab. Sorry, I should have had that publication here. The sequence identity between MSA and the triple mutant streptavidin that we were using is actually very high, 57%. If you overlay the structures, they overlay with a backbone RMSD of 0.5 angstrom, so they're actually very similar, as shown here. The overlay is so tight that it's even hard to see that there are two complexes here, both between the streptavidin and the biotin. We relax the 4NBX structure onto the MSA crystal structure, and you can see if you zoom in on the binding site that the biotin positioning and the anchor point to position 103 in the CDR loop are virtually indistinguishable. So we had good confidence that the switching over to MSA at this stage would not be a problem. Okay, so does this work? Let's look at some control experiments so we're all on the same page about how we're doing the measurements. So we're using surface plasmon resonance to measure binding affinities in a B core instrument. We've looked at multiple configurations of the experiment where we either immobilize biotin on the B core chip or immobilize the MSA protein on the B core chip. For all of the subsequent experiments that I'll show, we've utilized the MSA immobilized on the B core chip. But we're always comparing to the binding of MSA to biotin with the biotin immobilized on the chip. And we've just done that for experimental convenience. Okay, so here's a sensorgram for MSA wild type binding to biotin immobilized on the chip. This is, excuse me, one specific example. It's a fairly robust experiment. It yields in this particular sensorgram, the fit yields a KD of 6.9 nanomolar, running it in triplicate with 3 biological replicates. We end up with a KD of 7 nanomolar, which is consistent with previously published values of biotin bound to this monomeric streptavidin. We also wanted to make sure that there wasn't any significant difference with biotin binding to an immobilized MSA on the chip. So we biotinylated the N-terminus of a sumo protein. This particular sumo protein has a very free N-terminus, suggesting that there would be no interaction between the biotin and the protein, and we'd just be looking at a biotinylated protein binding to MSA. And as expected, we get very similar binding interaction in this particular sensorgram, 7.9 nanomolar, so virtually indistinguishable from the experiment where the configuration is reversed. As I'm sure many of you know, surface plasmon resonance experiments tend to have trouble with distinguishing, extremely tight binding, and so we wanted to create a version of the monomeric streptavidin that was compromised in its binding ability to biotin, so that we would have a lot more room to see improvement with the binding of our molecules. So we introduced a point mutation within the biotin binding site in streptavidin, a serine to an alanine mutation that significantly diminishes the binding interaction to biotin and drops it to just over 1 micromolar. Again, we get, very clean data for the surface plasmon resonance experiment. Okay, so what happens with our design molecule? So this sensorgram shows wild type nanobody, its interaction with immobilized monomeric streptavidin. And as expected, and we observe that there is no interaction. So the nanobody by itself does not bind streptavidin up to concentrations of 100 nanomolar, despite the fact that the binding interface shows some similarity to the native antigen biotin or native antigen antibody complex. So that's good. If we now look at, here's our control of MSA bound to immobilized biotin, 7 nanomolar interaction, and then our biotinylated or nanobody at the design position of 103 gives great data, and in triplicate generates a KD of just below 2 nanomolar. If we look at our binding compromised monomeric streptavidin, recall that it has a KD of just over a micromolar. Binding of our design molecule to MSA S27A also shows significantly improved binding, resulting in overall roughly a 4 to 5-fold binding enhancement for the base design. And so the question is then, can we optimize the binding interface to get to a better binding affinity? So we employed our standard computational protein design algorithms to look at designing CDR positions for enhanced binding. We selected 2 variants, V119, which was our highest ranked variant by energy. And then we also selected a second variant, V149, which gives the highest number of predicted hydrogen bonds. And we were interested in this configuration because we were very interested in determining whether we could actually tune the kinetics of the interaction between the antibody and its target by off-rate optimization. So the hypothesis is that if we build in a lot of hydrogen bonds, the off rate should go down and we should be able to get tighter binding because of that. So both of these variants are 4-fold mutations, 2 mutations in CDR3 where we have the biotin attached, and 2 mutations in CDR loop 1. And you can see that they we're going to do some new interactions, and in particular for variant V149, we're introducing a number of new hydrogen bond and polar interactions. Okay, so we evaluated these experimentally in a similar manner. Those data are shown here. On the upper left, again, is the control experiment with just biotin binding to wild-type MSA, 7 nanomolar interaction. Our first round design of sort of the naked, unoptimized interaction, unoptimized nanobody binding with 2 nanomolar affinity. And then for variant 119, we get roughly a 2-fold improvement in binding affinity, but for the sort of off-rate optimized variant, we get roughly an additional 15-fold improvement in the binding affinity. We're now down to 100 picomolar, which we thought was a great result. Second round of optimization, we get between 2 and 15-fold improvement of binding, including achieving the target off-rate enhancement. So, we get basically roughly a factor 2 improvement in the off-rate relative to the starting molecule, which was gratifying. Unfortunately, things were looking like they were working too well. And then we analyzed the molecules by size exclusion chromatography. We observed that our best variant shows aggregation. the parent of 149, V119, or sorry, the first second generation design, V119, is cleanly monomeric, as shown in this SEC trace. Unfortunately, sorry. Right, so unfortunately for our best variant, 100 picomolar binding, it's clearly aggregating. Here's where the monomeric peak should be coming out. There's a considerable amount of aggregation. And how am I doing on time? Okay, so right, so we attempted additional CDR optimization on the 149 molecule, added 6 additional mutations at various locations in the CDR to generate a variant called V186. That variant, unfortunately, shows even greater aggregation. It still retains binding, but the aggregation problem was exacerbated. So we decided to try to evaluate what was happening with the aggregation by using MD analysis. And so this is a 100 nanosecond MD simulation of the nanobody bound to MSA, focusing on CDR loop 3. And what you'll see is that when I start the run, this CDR3 loop will actually unwind a little bit and lose its contact with the rest of the protein framework. And you'll see very quickly the loop will extend into a conformation, although it's hard to see from this view, sort of unpacks itself from the rest of the protein. So analyzing the MD simulation, it suggested the introduction of 2 additional mutations, but these mutations would be in the framework: an alanine to valine mutation and a phenylalanine to tyrosine mutation. So that double mutant on top of the prior mutations generates a V186 generates a variant called V186 framework. And then this is just showing some molecular detail of what's happening with those mutations. If we look at comparison between solvent-exposed surface area for the parent V186 versus the parent with the 2 framework mutations, what we see is that for 2 different hydrophobic clusters on the nanobody, that the simulation generates or shows that the double mutant shown in sort of red has fewer snapshots of exposure of the hydrophobic clusters to solvent, suggesting that the V186 framework mutation variant should be less prone to aggregation. And we actually observed that experimentally. So the compared to the 149 parent that I showed previously, which had lots of aggregation, the framework mutations rescue significantly the aggregation tendency of the variant. There's still some stuff happening, but we're getting mostly monomer. And satisfyingly, that variant still binds with very low nanomolar, high picomolar binding affinity. And so the variant, this framework variant binds with 35-fold greater binding affinity than biotin alone to the MSA wild-type target, and 21-fold tighter binding affinity to the compromised streptavidin target. I haven't shown the data for this, but it's similar. It's similar in quality to the other SPR traces that I've shown. So to summarize, CDRs, Cdr Extended Antibodies are natural evolution to the antibody-drug conjugate concept, offering potential for the creation of a new class of protein binding molecules. The computationally driven design of CDR extended antibodies can be achieved through a systematic, stepwise approach that leverages well-established computational protein design methods. In this demonstration system, this model system, we were able to show the viability of the design concept, and we were able to achieve greater than 20-fold improvement in binding relative to biotin alone, starting from binding affinities as weak as a micromolar to as strong as 7 nanomolar. And perhaps the most important information is who did the work. The work was primarily done by graduate student Jingzhou Wang, who's a graduate student in our biochemistry and molecular biophysics graduate option. Jingzhou had help with some of the computational work, particularly the MD simulations, by Adenylate Kinase, who's a graduate student in our bioengineering option. Both tremendously talented graduate students, and I'm really thankful for their joining the lab and working in the lab during my hiatus as division chair. Also general support from Monica Bretkow for laboratory operations, and support from the Caltech Protein Expression Center, which is directed by Joost Wilmetter. And with that, I will be happy to take questions.
Okay, great. Thank you, Steve. Do we have any questions? Okay. Yes, there's a question here, Steve, for you.
Am I, let's see, how do I get out of sharing on this? There. Did I break anything?
No. Can you, so Steve, can you click on ask a question so that you can see the question and you can answer that?
Okay.
The one at the bottom.
Is there value in considering that one?
Yes.
Is there value in considering potential interactions between your tethered small molecule and your nanobody and designing against these? Negative design. In principle, these could interfere or compete with the desired interactions between your nanobody and its target. So we definitely, the design calculations certainly include the small molecule as tethered to the nanobody. So all of those interactions are completely included in the design calculations. I'm not necessarily understanding what the negative design implication is there. But it is certainly the case that we include the small molecule in all of the calculations. Let's see. So there's a question: did the cysteine that you use for conjugation pose problems in some cases? So we tested the nanobody with just the cysteine introduced at the particular CDR position, and those molecules were still well behaved. So in general, the introduction of the cysteine didn't really compromise the stability and solution properties of the nanobody.
Steve, maybe I can ask a question.
Please.
So I was really interested in your docking protocol. So can you give more detail? For example, did you use trials to do this? And if so, how did you define the binding interface?
Right. So the docking was done using a fast Fourier transform docking algorithm that was developed years ago. I don't have the exact reference off the top of my head, but it's a in those cases we were using sort of fixed loop configurations derived directly from the crystal structures of the nanobody antigen complexes, and doing rigid body docking, and then within Triad, which is our computational design optimization or optimization software, allowed for relaxation after the initial docking. And then the scoring, the interface metrics were all computed using literature methods, but implemented it within the Triad software package.
Great, thank you. You have 2 more questions.
Yeah, Chris Snow points out the FT, that is the FFT docking method that we're using in the comments. Thanks, Chris.
And there are 2 more questions and answers.
One question is, how would you estimate the benefit of using deterministic algorithms instead of Monte Carlo and continuous rotamers instead of rigid rotamers, especially on smaller design problems? Great question. Obviously, Monte Carlo is a stochastic algorithm. It's not deterministic. And that's one of the downsides, is that you're not really guaranteed to get the solution. However, for designs of this level of complexity, which I would say are relatively small because we're only looking at a design of a small region, we're probably, we can be reasonably confident that we're actually getting good sampling and finding the ground state solution. We could use, because of the problem size is relatively small, we could use deterministic algorithms. We just haven't done that, but it would certainly be worth doing. On the question of continuous rotamers versus rigid rotamers, that's a great concept. I've thought about that for many years, about how to implement a continuous rotamer concept, and then how to match that with a common curl optimization approach. I was never able to come up with anything tractable. If you have any great thoughts on that, I'd love to hear them. Yeah, I think that would be really, really useful if we could, for the field, if that could be implemented. Roberto says that Bruce Donlan has developed methods for this, which would be great to, I should go look at that. Thanks, Roberto. And another question is, can the computational design approach, or accommodate alternative chemistries, such as non-canonical amino acids, enzyme-mediated conjugation strategies, or other conjugations with canonical amino acids? The answer is definitely yes. It's yes at multiple levels. So there's a question of force field, obviously. So if you have different types of molecules than just amino acids, we can handle that because we tend to use the Dryden force field, which covers, a near infinite spectrum of molecular interactions with parameters, but with systematic parameters. And then, of course, the chemistry that you want to use is the chemistry that would be the most convenient for your approach. For the model system that we selected, we selected the Biotin Streptavidin system because the conjugation chemistry was really straightforward. Just coupling the cysteine using well-established protocols. More complicated situations, I think, will obviously will require different types of conjugation chemistry. We're very interested, for example, in looking at rescuing antibiotics that are no longer useful for treating, multi-drug resistant Staph aureus, for example, and rescuing those antibiotics by adding an antibody to build a tighter binding interaction and allowing for interaction with PBP2A, for example. In those cases, the coupling chemistries that we were looking at are much more complicated than using, cysteine and maleimide. So all of those things can be accommodated within our computational framework. There's a question in the chat, which is, was there any testing of the Cdr extended antibody against similar target protein surfaces to determine off-target binding? We have not done that yet. One way we could approach that would be to look at surface mutations, even by creating surface mutations on the streptavidin itself, that one might imagine would interfere with the binding of an antibody to the design interactions with our antibody streptavidin target. That would be something interesting to do. We haven't done it yet, but that would be an interesting thing to do, and perhaps even to do more broadly, to look for whether the biotinylated antibody will bind sort of other targets. Another question, have you considered using experimentally derived ensembles of each antibody conformation prior to docking so as to increase sampling of the binding site? That was from Roberto, a great idea. We have considered that as a general matter in our design approaches. We haven't done it specifically for this case, but it is certainly something that we could easily handle. So the idea obviously would be to take an antibody structure, apply, for example, a short run of MD simulation to generate a structural ensemble, and then to look at each member of that ensemble in the FFT docking algorithm, which is fast. So it wouldn't necessarily increase the pose generation time by that much, and could potentially give a lot more choices for initial poses. Another question is, were any other docking pose metrics considered other than distance and surface area? Yes, there were there were four things that we looked at to determine which poses were most desirable. Let me go back to that slide. I won't share it, but I'll just, so there was interface separation distance, buried surface area, a shape complementarity criterion, and then the fourth thing, well, I guess there were two additional things. One was that the pose had to cover the biotin binding site, and then the final thing was that we really wanted to see examples of specific interactions in our poses that recapitulated the nanobody native antigen binding interactions. And you may recall there was a slide I showed where for the pose that we selected, two interactions that existed in the native nanobody antigen complex were also recapitulated in our modeled nanobody streptavidin complex. Questions from YouTube. Thanks for the great seminar. I'm curious to know if you can predict in silico that the proteins will aggregate or any other unfavorable property. Could this be built into in silico selection? So, so I think the answer is yes, and evidence for that is the the MD simulations that I showed on a particular variant and a variant's sort of framework mutations clearly showed differences in the exposure of hydrophobic surface area between those two variants, with the idea that hydrophobic surface area exposure leads to aggregation. We also have other work that I haven't talked about here at all that Aiden Estevez is working on, looking at solution properties of insulin and insulin variants. And we can also see, based on MD simulation, we can generate predictability about the solution properties of various insulin variants. So I think in general, there is great promise in utilizing MD simulations and other computational approaches to predict solution properties.
Okay, great. I think we're out of time, unfortunately, but that was great, Steve. Thank you for the nice presentation. Very exciting to see what your lab has been up to. And thank you, everybody, for asking excellent questions and for your participation. I hope to see you next time. So again, thank you, everybody, and thank you to all the speakers.